So, one final presentation on it's also methodology related. It's from genetic analysis about measure of gut bacteria diversity. And Graceline and Tina will present. Yeah. Please.
Thank you. Yeah. So first of all, thank you for inviting us. And I take this opportunity to present about our company, Genetic Analysis, and also, introduce about our test, the GMAP dysbiosis test, and then briefly go through the 2 new tools that we have added to our reports. So we are a molecular diagnostic company that is based in, we are a molecular diagnostic company that was founded in 2008. And this was based on a research that was done by Professor Knut Rudi and is patented by the Norwegian University of Life Sciences. So our test, the GMAP dysbiosis test, is it uses fecal samples to profile the intestinal microbiota using the 16S rRNA gene. The aim of our test is to identify and characterize dysbiosis in IBS and IBD patients. And in our test, we have a selected markers that we target. And these markers are selected based on its ability to distinguish between a healthy gut and a dysbiotic gut. And as we know, if we need to make such distinctions, it's very important to characterize the microbiome of the healthy individuals. And researchers have identified that there are stable patterns of bacterial signatures that are found generally in all healthy individuals. So any deviation from this healthy gut microbial patterns is often referred to as dysbiosis. So in order to predict if a given sample is either close to a healthy gut or a dysbiotic gut, we have developed the GMAP dysbiosis test. So here is a brief overview of our test. We extract the DNA from the fecal samples, we amplify the 16S gene, and then we detect only the selected bacteria markers. And then the markers have a luminescence attached to it, and it's read by the Luminex machine, which is then fed to the software, which generates the report. And the entire process takes about just 2 days. And in the 16S gene, we focus on regions from V3 to V9. We have the 48 marker panel, which is our standard panel that we use, and it's also CE marked. But we have 2 other panels that we generally use for research purposes. And it's important to note that we have compared the relative abundance of the bacteria markers that we get out from our platform to the sequencing platform, and we have illustrated that it has quite good correlation. So a little bit about our reference model. So we have developed a reference model that's a normobiotic or healthy gut profile model. And this model has been tested and validated for its ability to differentiate between a healthy gut and a dysbiotic gut. So when a sample is fed into the software, it algorithmically compares the abundance of the sample to the reference model, and a simple and easy to interpret report is being generated. So we can sort of subdivide the report into 4 major components. So the first is the dysbiosis index. So we score the sample as either from values from 1 to 5. So if the deviation of the sample, input sample, is quite less compared to the reference model, then those samples get a value of 1 and 2. But if the deviation is much larger, then those samples get a value of 4 and 5, and we refer to them as dysbiotic samples. And we also, it's important to remember that this index algorithm mostly depends on the presence of the commensal bacteria in the gut. The next, we also report all the relative abundance of the bacteria of these 48 markers that we have. Then we also look into the diversity of the sample. And one important difference is that where diversity looks at the overall diversity that's found inside in the sample, the dysbiosis index is more based on the commensal bacteria. And here, and we also look into the key bacteria that are found in the gut that maintain important functions in the gut. So these are two new tools that we have recently added to our report, and so we'll briefly go through each one of them with an example. We estimate the diversity of the sample, and here in this example we have a healthy IBS and an IBD cohort. So in the y-axis, so when the algorithm calculates the diversity, it outputs a score from anywhere between 0 to 5. And lower the score, it means less diversity in the sample. If the score is higher, then it means it has higher diversity in the sample. So here in this example, we can see the diversity score in the y-axis, and the healthy samples have a quite higher diversity as expected when compared to the IBD samples. And the IBS samples have more or less comparable diversity as the healthy. So now if you wonder how the diversity and the DI correlates. So here in this plot, the x-axis is the dysbiosis index, and the y-axis is the diversity. So the green circles, the healthy samples, as we would expect, we see that they have a lower dysbiosis index and a higher diversity. And the IBD samples in purple, we see have higher dysbiosis index and then lower diversity in most cases. And the IBD samples are more or less spread across. But here in this right corner, we have these IBD samples which have higher dysbiosis index, but also have a higher diversity. So this actually is because our algorithm that calculates DI mostly looks into the balance of the commensal bacteria rather than the diversity. And the next is the functional bacteria profiles. So here, based on literature search, we have subgrouped our markers into different profiles based on the function. And these are major gut functions like the butyrate producers, the gut mucosa protectors, etc. So here are the selected markers for each of these profiles. So we apply a certain criteria to these profiles based on the abundance data that we have. And if all of these markers would satisfy the criteria, then the profile is reported as balanced. But if these markers do not satisfy the criteria, then the profile is reported as imbalance. So here in this example, I've used the same cohorts as I used before, And on the x-axis is the groups, and in the y-axis is the percentage balance and imbalance. And here we can see the healthy samples have a much lower imbalance profile. The imbalance is shown in dark orange. And the IBS samples have a slightly higher imbalance in the gut. And IBD samples have even more imbalance in the gut. So this is a very quick way for the clinicians to just get a peek into the gut functions of the patients. So next I would just like to illustrate how we could use the signal data that comes out from our software to compare, in this case, monitor treatment progression over time. So here we have looked at 40 C. difficile patients before and after treatment, and we compare them to healthy cohort and IBS cohort. And this is an usual PCA plot, and here are those samples, the C. difficile patients before treatment. And we can see that the gut microbiota of the C. difficile patients are quite different from the healthy, shown here in green, also from the IBS, which is shown here in orange. But after 8 weeks of treatment, we can see that these these individuals that have undergone the treatment have their gut microbiota now much closer to the healthy and to the IBS. And it's good to note that the IBS samples, we see quite a large variation in the cohort. And this is a general trend we see with IBS patients, where the gut microbiota is sometimes, in some cases, quite similar to that of a healthy, but in some cases quite deviated from the healthy. So apart from clinical diagnostics, we are also involved, we have also partnered with NMBU in developing resources like the HumGut. And this is a database that is a collection of the most prevalent bacteria genomes that are associated with the healthy gut. And this database can be used as a reference genome reference for all human metagenome studies. And in future goals, yeah. So we know there are quite a bit of challenges when it comes to microbiome-based testing, but we see that microbiome-based diagnostics are the future. So recently we have got financial support from Norwegian Research Council, and this is for a project where we intend to develop a non-invasive test of for the IBD patients that would predict the disease course and also provide guidance to identify optimal treatment options. And this will be carried out in collaboration with university hospitals and our research and development partners. So we believe this would create new opportunities for developing new products that will be utilized in the future in other disease areas. So thank you, and here is our team. Yeah.
So, thank you very much for a nice presentation. Question, Fredrik?
Hi, Fredrik again. And again, I have two questions.
Okay.
Firstly, I was wondering, these C. diff patients,
Yeah.
what kind of treatment did they receive?
They have been through two different kinds of treatment, but I'm not exactly sure if I can say, but it was some sort of probiotics.
Okay.
Probiotic treatments.
Okay.
Yeah.
Because I think it would be very interesting to know if there was a difference between those who got fecal microbiota transplantation or antibiotics.
So in this study, they did not do any fecal transplant.
Okay.
So this was just probiotics. So two different kinds of treatments they've had.
Yeah.
Yeah.
And then the second question is, did you look at any subgroup analysis in the IBS group? Because you mentioned that there was so wide diversity, but could it be related to some of the subgroups?
It could be, but as I mentioned, we have very selected bacteria markers. So we have not looked into that. Yeah.
So I have a question. So you have developed this now with some diversity measures. I was wondering, we typically when we talk about alpha diversity, we talk about richness or evenness. So is this like, is there more different bacteria, or are they more evenly distributed? How does this diversity measure relate to typical diversity measures like Shannon or
So
KO1?
we have based us mostly on the Shannon, but we had to develop our own algorithm because this is based on signal data rather than the sequencing. So this is more based on both evenness and species richness. Yeah.
Do you know how it would correlate to if you have you investigated your samples also, for instance, using cysteine sequencing, and then to see how there is a correlation between that diversity measure and Shannon, for instance, based on cysteine?
No, not at the moment, but that's something we are planning to do in the future. Yeah.
Very good. Other questions? If not, I think thank you very much. I think time's up. Thank you very much for your presentation.
Thank you.
Okay. I guess we are perfectly on time then for a last 15-minute break before the final session.